Welcome to the Deep Dive. Today, our mission is to get you up to speed on what looks like a genuine revolution in imaging. For, 3 centuries, optics has been stuck with this optical triangle.
Exactly. It's a forced constraint. You want incredibly high resolution.
You have to accept a tiny field of view.
And you have to get extremely close. I mean, we're talking about putting a lens just 0.13 millimeters away from a sample to see the finest details. It's a physical wall.
A wall that seems to have just been broken. We're diving into the multi-scale aperture synthesis imager, or MASI, which researchers at UConn unveiled in January 2026.
And the core idea here is they've managed to decouple the physical act of capturing light from the synchronization you need for high resolution.
So they've essentially replaced the lens with software.
In a way, yes. And the result is just staggering. MASI is getting sub-micron resolution. They've validated it down to 548 nanometer lines, but from a working distance of 2 centimeters.
2 centimeters. Okay, let's unpack that. Why was this so hard before? We've had synthetic apertures with radio telescopes for ages. Why did it always fail with visible light?
It's what they call the wavelength barrier. Radio waves are long, they're easy to sync up electronically, but visible light waves are so incredibly short.
So the physical alignment has to be perfect.
Perfect is an understatement. You need to align the light paths to within 50 nanometers. That kind of stability is, well, it's basically impossible outside of a hyper-specialized lab.
Okay, so if you can't solve the hardware problem, the physical alignment, what did MASI do instead? What's the clever bit of kit?
They just stopped trying to solve it. The architecture is a distributed array of really inexpensive, independent CMOS sensors.
Independent, so they're not physically linked.
Not at all. And here's the hack. Each sensor is coated with this random phase mask. It's basically tiny microbeads, just 1 or 2 micrometers across, that scramble the incoming light.
Scramble it. That sounds counterintuitive.
Yeah.
How do you get a sharp image from a scrambled signal?
That's where the real magic is. It's the software. They call it computational phase synchronization, or CPS.
The software lens.
Exactly. Instead of physically stabilizing anything, the algorithm just treats the unknown positions of all those sensors as variables in a giant optimization problem. It computationally locks them together.
Right, so the software is correcting for all the physical jitter and misalignment in real time. And that gives you, a 3 times resolution boost over a single sensor?
At least, but the field of view is where it gets really wild. The system can create an effective field of view that's up to 16 times larger than the physical footprint of the sensor array itself.
16 times. So
Yeah.
what does that mean in practice?
It means you can capture a huge area in one go. For manufacturing inspection, for mapping cell cultures. It's just instant. No more stitching hundreds of tiny images together.
Let's ground this in some real-world applications then. Where does this change things immediately? You mentioned forensics, ballistics analysis.
Ballistics is a classic nightmare for microscopy. High-res lenses have such a shallow depth of field. A bullet casing is curved, so you only ever get a tiny sliver in focus.
And you have to manually stack images.
It's tedious and subjective. But since MASI recovers the full wave field, the amplitude, and the phase, it gives you a quantitative 3D map of all those tool marks.
And the key is you can refocus it after the fact.
Precisely. You capture the data once, and then you can digitally scroll through the focus. The entire curved surface is perfectly sharp. It changes everything.
That's huge for forensics. And I imagine the same logic applies to biomedical work. That 2 centimeter working distance you mentioned, that feels like an eternity in microscopy.
It is. It means you can image live cells inside a petri dish or a flask without ever risking contamination. You're not breathing on the sample.
Yeah.
And because it measures refractive index, you get what's called label-free visualization.
You mean like a virtual stain?
A virtual stain. You can see cellular structures without injecting potentially toxic dyes.
Okay, this is where we get a bit more visionary. If the algorithm can sync a loose array of sensors on a bench, could it sync a loose array of satellites in space?
That's the grand vision. They're calling it swarm telescopy. The same algorithms could, in theory, synchronize the light from a whole swarm of free-flying satellites.
You'd be synthesizing a virtual telescope.
A virtual mirror kilometers wide enough to potentially give us the resolution to directly image an exoplanet.
So the big takeaway here is pretty clear. Optics is shifting from being a hardware-defined field, limited by how well you can grind glass, to a software-defined one, limited only by computation.
And that leaves us with a fascinating thought for you to take away, because MASI captures the full, complex wave field. The next frontier they're looking at is non-line-of-sight imaging. using these algorithms to potentially see around corners. So if that pans out, what could that mean for search and rescue? For autonomous driving? Something to think about until our next deep dive.